Hello, my name is Carter White, and welcome to the guide to the Test Your Knowledge part of my website on cell-cultured meat. Before we get into how to solve the question and why the answers are the way that they are, we should discuss why this question is important. To start off, this activity is important because answering questions like these is something that geneticists are tasked with doing every day in this type of research setting. Of course, their work is to a much more difficult and extensive degree than the words and labels used in this activity. But the basic concepts of finding specific genes in DNA that codes for each part of the end product, no matter how large or small the impact, is what can help something like cell-cultured meat reach its fullest potential in taste, texture, and safety. This is also precisely why I chose to ask a question centered around this concept of finding genes related to tasty meat production in a mix of helpful and non-helpful genes. I feel as though it will give you a good idea to the type of work geneticists have to do, even if my task is a very oversimplified version. I also think it'll provide a decent foundation for those of you who are thinking about exploring the idea of studying genetics, biology, or really any type of human or animal sciences more deeply in the future. This idea of genes being turned on and off and sort of on or sort of off to different degrees does not go away in those fields. So again, that application of digging and sorting through this large-ish amount of genes in order to find the right ones or the best ones should help those of you looking into this type of career path. And it's hopefully not too boring for those of you who are not. Maybe you'll even find it interesting. All right. Now that we know why we're here, let us get into answering this thing. So the question reads, in this activity, a list of potentially important genes, tissues, cells is provided below that could or could not be useful for turning a set of DNA into edible meat. Please select and write down which of these 9 options would be useful in the culturing meat process. And then it lists myoblast instructions, adipose instructions, Peter-Lie instructions, neural cell instructions, Rachis instructions, endothelial cell instructions, connective tissue instructions, calamus instructions, and thyroxine instructions. All right, the correct answers are listed here as myoblast instructions, adipose instructions, neural cell instructions, endothelial cell instructions, and connective tissue instructions. Now I'm going to go through why each of these answers is correct, and then on the next slide I will walk you through why the other answers were incorrect. So myoblast cells are muscle cells. So of course they are very important to producing tissue that is primarily muscle for consumption. When we eat meats, most of that meat is in fact muscle cell. So this would be the main component, very important. We should have these instructions. The next one, adipose instruction. Adipose is basically the fancy version of fat cells. Fat cells are important in the complex matrix that makes up that chunk of meat that I talked about earlier in the website. It's very important to how the meat tastes, as well as adding that steak texture. Neural cell instructions. Neural cells are the cells in the nervous system. They aid, again, into the matrix of meat tissue and help replicate that standard farm-grown meat we know now. So it's another one of those little details that these muscle cells and these muscle tissues that we eat as meat, they contain these neural cells, because if they were in the living cow as a whole organism, those tissues would have neural cells. So we should add it into the meat so that it is consistent with that texture and taste that we are comfortable with now. Next is endothelial cell instructions. Endothelial cells are cells that line many of the vessels in an animal's body, including humans. This includes blood vessels mainly. Endothelial cells help make up meat tissue by mimicking the natural existence of blood vessels in animals. Again, this is another one similar to the neural cell instructions, where endothelial cells are within those blood vessels, which connect to a lot of the tissues in any organism, really, any animal. And you may have noticed maybe when you're cooking a steak, if you've ever seen anybody cook a steak or chicken, there might be blood on the grill or on the meat itself. And this kind of hints at the presence of something like endothelial cells to provide blood to this tissue in the animal. The last correct answer in this activity was connective tissue instructions. Connective tissue is part of almost every tissue, and as kind of the basement foundation, it helps tissue stay together and anchored wherever it exists in the body. Connective tissue supports and protects tissue and is therefore very important to add into the matrix of cells that make up meat. So this is another one that helps with the consistency and the feeling we get from normal traditional meat. So it's important for this cell cultured meat. The incorrect answers start with peterile instructions. This peterile is the confined and precise area in which feathers are found, which is where smooth muscles attach to feather follicles and can change and make feathers stand up and or relax. And it's not very important to the meat or meat matrix, as it normally indicates an area of skin that is associated with the movement of feathers. This doesn't have a lot to do with the meat that we eat. We don't really care about making feathers move in this area of skin. Normally meat is skinned anyway down to just the muscle. So this is not particularly important to add in our culture. The next incorrect answer is rachis instructions. The rachis is the shaft of the feather. It's four-sided tapering elastic shaft that runs the length of the feather. This, of course, is not very tasty or edible, as it is the hard part of the feather itself, that part you can hold on to, and gives it some structure. It's not important to make meat that is intended to be eaten. If you had these in your meats, then it'd be cutting up your mouth and poking you all over the place, and not very comfortable, not very important. The next incorrect answer was calamus instructions. Calamus is the quill, or the lower shaft of the feather. You might recognize quill from the olden days when people would use quills and ink to write. So obviously this won't be very important, but it's the round, hollow, transparent part of the feather tip and contains some conical scales. So again, it's not very important to the matrix, and it's not very important to the structure or taste of a piece of chicken. Although this feather tip is a lot of the times where the cells themselves are sourced from the chickens. This is an easy, non-invasive, non-harmful way to gather cells from a chicken. It's just take a feather that fell off and cut off the tip and take some of the cells on the tip. The last incorrect answer would be thyroxine instructions. Thyroxine is a hormone in chickens that helps the differentiation of feathers by increasing activity of feather-forming cells. This is not important in differentiating meat. It is important in differentiating feathers, so it is not important to the meat or the meat matrix. It's also not even a cell. It is a hormone, a chemical, so it would not be that useful and not important to add to our meat. Thank you for participating. I will encourage you to head back to the site for more information on benefits and innovations in this new and exciting and emerging field of cell-cultured meat. Thank you.